In 1996, biology was a stubbornly physical science. It was defined by hours of repetitive manual labor at the bench, mixing microscopic amounts of clear liquid just to observe the natural world. Reading the genetic code meant developing sheets of radioactive film and squinting at stacked, ladder-like bands just to manually transcribe a few dozen base pairs of DNA at a time. Even the digital side was highly constrained. Early genetic databases like GenBank were mostly collections of short, disconnected scraps of code, frustratingly incomplete puzzles rather than a cohesive library. Writing DNA was even harder. Commercial synthesis required massive, chemical-guzzling machines that realistically could only stitch together short strings, usually maxing out at 50 to 100 base pairs. Imagine taking a specific scientific paper published in August 2026, dropping it through a wormhole, and landing it on a 1996 computational biologist's desk. To a scientist operating within those 1990s constraints, the contents of this document represent a hard pivot. The work described here treats biology as a generative engineering discipline, where complete organisms are designed from scratch on a screen. The paper in question, published by Samuel H. King and his team, outlines how they used an artificial intelligence to design 302 complete, functional virus genomes from scratch. Researchers trained the AI on over 2 million bacteriophage genomes, data that would instantly crash a 1996 server. While 1996 software simply hunted for exact matches of mystery sequences in nature, this model doesn't search. It digests data to deduce DNA's structural rules. This allows researchers to perform de novo generation. They can sit at a prompt interface and instruct the AI to write a fully formed, mathematically valid DNA sequence that has 0 exact ancestors in the natural evolutionary tree. The software has absorbed the complex grammar of life so thoroughly that it can fluently author entirely original code on its own. But a digital text file of A, C, T, and Gs is just a blueprint. The next hurdle is pulling that AI-generated text off the screen and building it in the physical world. Recall that in 1996, printing 50 base pairs of DNA was a tedious, expensive chore. The viruses designed by this AI required continuous strands of 30,000 to 50,000 base pairs each. The researchers took the text files for their 302 custom viruses, emailed them to a commercial synthesis lab, and had those massive genetic strings chemically printed into physical existence. This brings us to the boot-up problem. You can print millions of atoms in the exact shape of a virus, but the ultimate test is whether that inert chemical string will actually boot up, hijack a host cell, and operate as a living, replicating entity. Out of the 302 digitally hallucinated sequences they printed, 16 successfully booted up into infectious, fully operational bacteriophages on the very first try. There was no manual tweaking required. No human scientist stepped in to rationalize the gene spacing or fix a folding error in the protein structure. An algorithm independently composing a novel biological blueprint that seamlessly boots into a living entity marks a permanent change in what is possible in a lab. At this point, the mechanical awe starts to give way to a colder reality. We are bypassing the traditional filters of the natural world. For the entire history of modern biology, our ability to innovate was strictly bound by our capacity to understand. This 2026 paper proves that those two tracks have now been completely separated. We are now operating within a black box model. A neural network can output a fully functioning, highly complex pathogen, but it cannot explain the underlying physics of why the organism survives, and the researchers utilizing the AI do not need to understand it either. When generating novel life no longer requires deep biological comprehension from the user, the expertise bottleneck that previously slowed biological development vanishes. The traditional safety focus on regulating specialized laboratory knowledge loses its primary point of friction. Humanity has crossed a threshold into programming novel life into existence, effectively authoring biological code that we do not yet have the capacity to comprehend.